Good morning, good afternoon, maybe good evening to some people. Thanks very much, Marianne. I want to firstly say thanks to the organizers, Caroline, Marianne, thank you for inviting me to give the webinar today. It's a real pleasure and a privilege to do that. So let me get my slides up. If I don't hear anything, I'm assuming people can see my slides. Okay.
Yes, your slides are visible.
Thank you very much. I take it as Marianne said, we're going to do questions at the end, so I can't see any hands, so we'll just do that in the end. Okay, so the title of the webinar today is the mRNA Technology Platform Development for Low Middle Income Countries, and I'll tell you a little bit about this from the Afrigen perspective. Okay, so the presentation today, I'm going to cover it in 3 parts. The first part is going to be a little bit about Afrigen's journey and where we are today. The second part is going to be focusing on mRNA technology platform that we've developed and the transfer to our low middle income partners. And the third part is where I'm going to show you our strategy for validation of the mRNA technology platform and the pipeline for sustainability. Okay, so Afrigen has had quite an interesting and unique journey. We are a relatively young biotechnology company. We were established in 2014, 2015, but only really became operational in middle of 2016. That's when I joined the team. And the mandate for the company at the time and continues to be today is to focus on capacitating manufacturing of vaccines in South Africa, in Africa. And so together with our partners at the time, our shareholder, the Industrial Development Corporation, and the Infectious Disease Research Institute, which is a non-profit organization, recently been renamed as AHI. We established the first adjuvant formulation center in Africa, actually the first one in the Southern Hemisphere. And together with the partners and BioVac focused on development of their TB vaccine called ID93, formulated with a adjuvant called GLA-SE. We quickly realized that in order to make the company sustainable, we needed to look at ways to diversify. And so the adjuvant platform that we established we used the formulation capabilities to extend that to other wellness and health products. And in terms of the formulation capabilities, we have built strong expertise in nanoemulsions and liposomal formulations. Our work on the adjuvant platform has really been through collaboration because adjuvants go with vaccines. And so we establish an extensive network with those that are developing vaccines, with those who have antigens. We also explored the immunotherapy landscape at the time, but during the business plan, we realized that you need to have a considerable basket of biosimilars, for instance, if you want to take that forward and have a bankable business. Then moving forward, we started to construct our first R&D laboratories. So prior to the construction of our own labs, we were renting facilities at the South African Medical Research Center, so very small laboratories. And at the height of COVID, managed to convince our shareholders that we should build our own labs. And this started in 2020. During lockdown, even the contractors had to have special permits. And then a very pivotal part of our journey thus far is when we were contracted by the WHO along with other partners to be the global mRNA technology transfer hub for low middle income countries. And we began the development of the mRNA tech. At the time, when the project was conceptualized, there were great hopes that we would have a technology transfer from one of the commercial companies like Pfizer or Moderna, or even some of the companies that had mRNA vaccines already in the clinic. Due to many reasons that didn't happen. And so this project became a greenfields project. And so we had to pivot a lot of things. We had to redirect staff. We had to redirect parts of the facility that had been sort of earmarked for other activities. And we started the real facility construction around having a GMP vaccine facility, and we envisage that we will have this licensed in the middle of next year. We also had to grow the people quite significantly, and I'm going to talk a little bit more about that later. So we really see our role in the biotech landscape around vaccine development. So we've been working with partners in developing various diseases, especially those that have high impact in Africa. These include TB, HIV, RSV, Rift Valley fever, gonorrhea, and COVID-19. And the aim is really to address health challenges, but also to address the underrepresented challenges in global research. And building local manufacturing capacity, as I said, this was at the heart of Afrigen when it was started, and we aim to support vaccine production capacity and really promote tech transfer, skill building, infrastructure development, so that we have a self-sustained bioeconomy. And lastly, to continue partnering with global organizations, global suppliers, because we have to secure the supply chain, to work with partners in terms of research and innovation. And this is really exciting, and a message that we are driving along with many others at this stage is that Africa is not only a consumer of IP and innovation, that we are in fact contributing to the innovation space and creating new IP on the continent. Our facility and our company really is uniquely positioned in that we are able to do end-to-end research and development, as well as manufacturing under GMP conditions. As I said before, we are targeting our GMP inspection. This is with the South African Health Product Regulatory Authority towards the middle of next year. And all of the activities that happen in this space are underpinned with a quality management system. So it's really important that all your utilities are qualified. You have very specific material handling. We have our own quality control laboratories, so that we can do release testing for our products. And as I said, research and development, which is also end-to-end. I'll have a slide on that a bit later. Our GMP manufacturing suites, as well as all the quality control labs, which I mentioned earlier. And lastly, aseptic filling, so that you have, in the end, a sterile filled product that has been visually inspected, labeled, and ready to go out. So I decided not to do a video because I was very worried about technology failure this morning. So I've just got this static slide which shows you the GMP manufacturing facility in Afrigen. So we have about 2,000 square meters. It's not large. It's what we'd say sort of boutique manufacturing, but absolutely fit for purpose in the sense of being able to do clinical trial material for phase 1 and phase 2 clinical trials. Some of you may know that even on the African continent that we do not have capacity for manufacturing clinical trial material. And so Afrigen is uniquely positioned for this, but also the size of this facility and the amounts of finished product that we can produce also lends itself very well to making batches for neglected diseases and small vaccine batches. In the facility, we're able to do manufacturing in grade C, as well as grade B and grade A. So grade A is the highest level of clean room facilities, and that's where your product gets finished. We also have all the supporting facilities, which are, around being able to hold your master cell banks and working cell banks in a cryogenic facility, all the QC laboratories, which are on the side. This includes chemical, biochemical, and microbiology, as well as stability testing. So we do all our in-house stability testing. This helps us to ultimately define the shelf life of our product and obviously meet ICH guidelines on stability. Okay, so this picture just helps you to understand a little bit about the journey. So in 2021, this is what it looked like. This is what we were doing. We were putting up walls. So we've gone in a very short time from basic construction of the facility to full-fledged manufacturing suites. Here we are able to manufacture API, active pharmaceutical ingredient. In this instance, it's the mRNA drug substance. We are able to manufacture bulk drug product and also finished drug product. So in Africa, we have a lot of capacity for filling and finishing vaccines and other pharmaceuticals. We have a big gap when it comes to actual manufacturing of the API, and we don't want to be beholden to the greater north and higher income countries for the bulk product, because in the state of crisis in pandemic, we need to be able to produce vaccines ourselves, and so API manufacturing is critical. We also have an R&D center which supports our manufacturing. It supports all the development of the vaccine products. And so it includes things like making or designing your plasmid, which is the starting material. Also the ability to develop purification processes. We have a full-fledged formulation labs which center around adjuvant formulations as well as lipid nanoparticles in the case of mRNA. We have developed all the analytics in-house and have transferred that into our quality control laboratories. And we also have tissue culture laboratories. This is specifically for when we are looking at protein expression. It's one of the assays that we need to do to release the mRNA vaccines. So I know that there are quite a few of you who are interested in, what kind of careers or career development exists around this biomanufacturing vaccine development space. And so I've put in this slide just to give you a sense of that. Due to the WHO Medicines Patent Pool project around mRNA, it was a wonderful opportunity for Afrigen, but we had to grow our staff complement rapidly. So that meant that we had to grow our staff by about 300% in under 2 years. And so this graph shows you the growth. You can see male versus female. And very interestingly, about half of our staff all have postgraduate degrees. We have a range of different skills and different qualification backgrounds. We have in-service trainees and interns, which are really important aspect for Afrigen. It's part of our capacitating the biomanufacturing workforce. We have technicians, chemical and bioprocess engineers, scientists with various backgrounds, pharmacists and pharmacist assistants, and then various specialists. And so this graph over here shows you the different areas. Sorry, this one is slightly outdated, but it gives you an idea of where Afrigen employees are situated in terms of department. There's a very big, strong focus in manufacturing and obviously R&D. And quite recently, we have grown the engineering and validation team because this is so critical for ensuring that you have all utilities, facility, equipment qualified and validated. And then I've got a little breakdown here of our production team and our scientists. So in the production team, you have a range of backgrounds, and probably very interesting to you in our R&D team, our scientists have backgrounds in immunology, biochemistry, microbiology, biotechnology, chemical engineering, formulations. That's been quite a difficult one to find. We've had to build capacity in that space. But also we are very privileged to have purification scientists and structural biologists on our team. Okay, so that was a little bit about Afrigen itself, and now we'll talk a little bit more about this awesome mRNA technology platform development that we've been involved in and how we have been transferring our know-how and technology to our various partners. So I'm sure that you're all very well aware about the power of the internet. So in January of 2020, when the pandemic hit us, we had SARS-CoV-2 sequences that were posted on the internet, made available to 350 and possibly more labs worldwide who were using the sequences to make synthetic genes. So from the synthetic gene, we had development of vaccines using different platforms, mRNA and DNA. We had viral vectors being developed. We had protein subunit vaccines and adjuvants. And this gives you an idea of how long it took from the publication of those synthetic genes up until the time that it got into clinical trials. So in the case of mRNA, it was just 66 days. And that just speaks to the enormous power of the internet, a collaborative network of researchers and scientists working together, and that certain platforms allow you to move faster than others. And so there were very many people who were quite concerned about this rapid development. Like, why now, in the case of a pandemic, can we make vaccines so quickly, but outside of the pandemic could take such a long time? So some key points on this is that the unprecedented acceleration of vaccine development, particularly in the SARS-CoV-2 space, was because of years and years and years of research and development that happened in the space of MERS, SARS-CoV-1, and other fusion proteins. And that's helped researchers to really fast track the development. The other thing is that most of the risk also sat in the financial space. So there was a huge amount of investment that went into getting the development going as quickly as possible. And the third important point is that rather than doing things sequentially, as we would typically be doing, we needed to do development in parallel. So we saw that there were clinical trials that were happening in parallel, and that was also adhering to the fact that we were not taking any shortcuts when it came to authorization. And one last point I'll make on this is that there was a regulatory framework already established around emergency use. And this is why the two mRNA vaccines, Pfizer and Moderna, were able to get emergency use licenses early on, because that framework had already been established. And so in terms of the advantages of mRNA, we see that in terms of when we compare it to other vaccine platforms, I'm going to say it's relatively easy to produce. We have had challenges. Cost effectiveness, this one is still coming into question. Small footprint facilities, so that is really key. We have also seen that, you know, we're able to elicit humoral and cellular immune responses. There's versatility in terms of the disease target. And as we have seen with the COVID pandemic, we have seen that the safety profile is relatively good. So I've mentioned the advantages, which are really still opportunities to be grabbed at. But there are still challenges, and I won't go through this whole list. But some of the key things that come to my mind immediately is the utility of the mRNA vaccine, you know, being able to have thermostable vaccines. We have to think about getting the vaccines in arms of people. And when we have to have cold storage at extreme cold conditions, it makes it extremely difficult and impossible, actually, in many low middle income countries. We are still trying to understand the immunology around mRNA, fundamental structure function understanding. I've said that we need to address the temperature stability issues. But another important aspect is that we have to have capabilities that go beyond the CMC. So CMC standing for chemistry manufacturing control. So we need to understand host pathogen interactions in the case of antigen design and the vaccinology pipeline. And as we develop new disease targets, we need to ensure that we have functional assays to be able to test as we do the development. And then lastly, in the mRNA space, freedom to operate, particularly around lipids and so forth, is very convoluted. And we need to ensure that our partners in Africa and the rest of low middle income countries have freedom to operate. And we are working very hard on that. We also have to address the price of mRNA vaccines, and so that it doesn't become inhibitory. Okay, so we learned quite a few lessons in the COVID pandemic. And one of the key things is that where vaccines are produced matters. So if you look at this map, you'll see that in terms of global vaccine manufacturing, 73% of global vaccines are produced really by 4 companies in high-income countries. And 76% of global production by volume also happens in developed countries. So India is quite a good example of that. But when we look at what happened during the COVID pandemic, and we all know the story, that Africa, we were the last to receive vaccines. And so this correlates very strongly to the fact that we do not have vaccine manufacturing capacity on the continent. And so Africa only makes 1% or less than 1% of the vaccine it uses. And so there has been and is a strong drive to localize manufacturing. And so enters the mRNA program that was initiated by the WHO and the Medicines Patent Pool out of the COVAX initiative. And at the heart of the program was addressing access, addressing the fact that during a pandemic, we need to be prepared. And then we have to discuss what happens outside of pandemic. So we need inter-pandemic sustainability, and that requires R&D and innovation. And I'm going to talk a little bit more about this partnership network, but during the pandemic, we are able to collectively address the vaccine needs, particularly because we all have the same platform, and collectively we can make enough doses for the countries in the network. So the mRNA Technology Center is hosted by the South African consortium. It consists of Afrigen, of BioVac, and the South African Medical Research Council. Afrigen's role has been to establish the mRNA technology at a scale that is appropriate for phase 1, phase 2 clinical trials, and to transfer that tech to BioVac as the first partner and the 14 other partners in the network. And BioVac's role is to do further industrialization of that platform, to take it to phase 3 and to commercial manufacturing. We also need to sustain the manufacturing facilities by ensuring that we have a pipeline of vaccines that are coming through, and that we are improving the mRNA tech. And this is being done through an R&D network, which is being facilitated through the South African Medical Research Council. And here we are looking at other disease targets and second-generation technologies. We have strong support from the African Union, Africa CDC, and the Department of Science and Innovation. And as I mentioned earlier, the WHO and the Medicines Patent Pool have been co-leading this program. So the core objectives of the hub in Afrigen is to develop the mRNA tech to we've been using the COVID-19 as a proof of concept to demonstrate that we have comparability of the vaccine that was developed against an approved mRNA vaccine to transfer to the partners. And an interesting thing is that while we were doing all this and being on the ground, we were also able to evaluate the vaccine development value chain in South Africa and Africa and see where the gaps are, and to be actively involved in capacitating various research entities and organizations to try and close those gaps. And this includes things like being able to do GLP toxicology studies, being able to do challenge studies and so forth, looking at the supply chain, lots of different areas where collectively these all need to come together to have a complete value chain. So this mRNA technology transfer program really does have Africa at its center. We're building sustainable capacity and capabilities. A wonderful thing about this network is that it includes, or it straddles across 4 continents, as you can see here, and it connects 15 different countries by the companies that are represented here on the left-hand side. And this has a great impact because the potential is that we could reach 3 billion people through this network. And there was a lot of pressure on Afrigen as we were developing the technology to be able to package the learning and the know-how as we were doing the development. So we started by training the partners at very, very early lab scale. So by November of 2023, we had trained 14 of the partners that received lab scale manufacturing and testing at Afrigen. And where we are today, so by the end of 2024, the technology packages have coming to a completion, and we will have 4 of the 14 partners back in Afrigen to participate in the technology transfer demonstration and hands-on training at the larger scale. So quality by design, this is something that I think gets thrown out a lot as a phrase, and it's a little bit blasé, but it is so very important in our vaccine development space. So what you have here is a flow diagram describing the QBD approach to drug substance manufacturing process for a fusion protein. But this is applicable, you know, for other drug substances as well. So I've just used this here as an example. So abbreviations, they are defined here, but quality by design is a proactive approach to building quality into the product rather than relying on just testing, you know, testing the product to death at the end. So in the inception, in the design, quality is put first. And I like this phrase, which is quality cannot be tested in the product. It has to be built in by design. And so these are the typical steps that need to be followed as you go about developing your vaccine. And usually it starts by having a quality target product profile. You'll see that there are risk assessments scattered around in this process. It's a crucial part of this. And then the next step would be to look at what are the critical quality attributes that we need in our vaccine product. Other key elements are potential critical process parameters. And as you move along in the development, you come to tighter and tighter control, final critical control parameters, and so forth. And even with your raw materials, you need to develop a control strategy around that. And so in Afrigen, just to give you a sense of all the work breakdown structure that existed around this mRNA vaccine technology development, is that we've had this mRNA platform. We said, okay, so how will we ultimately validate this platform? Well, we can do it by having, you know, preclinical validation, but ultimately you want to see your performance in a clinical trial. And so in order for this to happen, we had to have all of these work packages done. And so that includes making sure that you have a GMP certified facility where clinical trial materials can be made. We had to develop everything from scratch. So it started out in R&D, and all of the experimental work, all the analytical method developments are very well described in development reports. We have to do stability studies, and we also have to have a strong preclinical package. All of these contribute to a clinical trial application. And once all the development has been done in R&D, it is transferred through a tech transfer from the R&D facility into the manufacturing facility. This very same package that is developed for tech transfer internally is what we use for tech transfer to our partners. So as I mentioned earlier, the quality target profile, product profile is the first step in your vaccine development process. Typically, it includes the indication for your vaccine, the type of vaccine, how it is administered, the dosage form, and so forth. And following on from this, you develop draft acceptance criteria for both your drug substance, in this case it would be mRNA, and then for your drug product, and that is the mRNA, which is formulated in the lipid nanoparticle. And so these are all the quality control attributes that we look at when we are characterizing and then releasing the vaccine product. And so, as I mentioned earlier, as you go in your further and further in your development process, where you have, you know, broader knowledge, you start to focus on all the design elements. So development, design, in-process controls, process validations. As you continue forward, you begin to have a much more controlled space. And an interesting part of this is this design space, which is something that is not very well established yet in Afrigen, but something that we will be moving forward in as we progress with different disease targets and as we take those forward into further development. So I mentioned earlier that we use the SARS-CoV-2 as the proof of concept product for the mRNA technology. And what you have here is a picture that shows you high level what the mRNA construct looks like. So it has your antigen coding sequence here in the middle, flanked by your 5' UTR and a 3' UTR. And on the 5' side, you have a cap. On the 3' side, you have a poly A tail. And these all contribute to the efficiency of which your RNA is expressed and translated in the body. In terms of, well, where do you start when you are going to make an mRNA vaccine? It is preferable and helps you to go a lot faster if you already know what the antigen is. So as I mentioned earlier, in the space of coronaviruses, there had been a lot of work done. And so the spike protein was identified as a good antigen to use. In the space where you don't actually know what the antigen is and you have to do antigen selection and so forth, it takes a lot longer. So once you have the antigen selected, you can move on to mRNA cassette design. And then following on from that, you're going to make your plasmid DNA, which incorporates all the design elements for your mRNA. The DNA is your starting material, so you will make more plasmid. And then following on from that, you'll go on to mRNA synthesis, purification, formulation, and finally finishing into vials. And an important aspect of this is that all throughout this process, you have to have analytics development and preclinical testing. And it really is an important and essential platform that has to be developed alongside the actual development of the drug substance and drug product. We are working on second generation products and technologies with our partners, and these are to address improving the potency and durability of mRNA vaccines, the thermostability, so that we can transport and handle it at more reasonable temperatures like 2 to 8, reduce reactogenicity. I said the reduction in cost of goods, so we need to make these vaccines cheaper. And we need to have freedom to operate. So to take you through the journey of scale up, so where did we start? We started at 200 microliters. So this picture gives you a sense of, the scale up process that we have taken on in Afrigen, starting at 200 microliters, and where we have landed up to now, which is at a 1 liter scale for the in vitro transcription reaction. We've been able to track the mRNA yield along the process, and it seems to be that you have a relative consistency in terms of the amount of mRNA that is produced. So what does the manufacturing platform look like? So when you're developing all of these things at 200 microliter scale, you also have to bear in mind, okay, how is this going to look like when I'm actually producing it at a large scale in a GMP environment? And so our manufacturing platform now has these various steps that are included. The manufacturing of the plasmid, which in Afrigen we are using conventionally, so using plasmid DNA. There are developments now, which are around using synthetic DNA, and we have some partnerships around that. Then you move into your drug substance manufacturing. There are several process steps in this. And while you are doing each of these steps, there are in process control tests that happen, and you're also characterizing as you go along. So you're checking what happens with mRNA yield, what happens with the purity as you go along the process. And then we produce the drug product, formulate it, and then it is, you have addition of sucrose, which is acting as your preservative, final filtration so that you have a sterile product, and then finish into vials. So in terms of our production capacity for clinical trial material, we can do 2,500 vials per batch. This equates to about 24,000 doses per batch. And so this is what we call our conventional process. There are other processes which are more automated, and we are also working with partners in that space. So as I mentioned earlier, the analytical profile is extremely important. We have developed 14 assays from scratch. So these are all of the ones that don't have this European pharmacopoeia or European USP, the American pharmacopoeia. So all the ones that don't have that, we have developed these from scratch. It takes a considerable amount of time and effort and human resources to do this. And when you're developing the assays, they need to be fit for purpose, but they also need to be qualified. So you need to show that they are reproducible, that there's linearity and so forth. The preclinical models that we have used to evaluate Afrivac 2121, which is our COVID vaccine. So in terms of immunogenicity, we have used the VOLP-C mice, Syrian golden hamsters. We are doing a characterization study in NHPs. And in terms of efficacy, we're looking at protective efficacy in the golden Syrian hamster model and in macaques. And essentially, and a prerequisite for going into your clinical trials is that you have to have a GLP toxicology study done. So this is a summary of all the testing that we've done on the batches. So from physical chemical testing to mice immunogenicity, looking at IgGs and neutralization activities. And as I mentioned earlier, your toxicology, which is a prerequisite. So just to say that on the Afrivac 2121, our COVID vaccine, due to the changing landscape for COVID-19 pandemic, and also due to, I want to say, a very small, almost negligible market at the moment, decision was made not to go forward with the clinical trial, and that the platform would be sufficiently validated using preclinical models. Okay, so that brings me on to the next topic. Sorry, I do know that it has been quite a long time that I've been talking, but I'm going to bring it to a close shortly. So just to tell you a little bit about how we validated the mRNA Tech platform and looking at the exciting future pipeline ahead. So our strategy was to really look at the preclinical comparability data. So of the vaccine in comparison to a commercial reference vaccine, we focused on, as I mentioned earlier, the physicochemical parameters. We looked at relative potency in vitro as well as in vivo. We also did in vitro immunogenicity to look at potential reactogenicity. Yeah, we worked with University of Pennsylvania to do this by looking at reactogenicity in human dendritic cells. We have followed a specific spike IgG antibodies. We've looked at neutralizing antibodies. We've also characterized Th1, Th2 responses. Also looked at cytokine profiles, and then in terms of efficacy, looked at protective efficacy in the hamster challenge model, and lastly, looked at the toxicology in rats. And so all of these have now been completed. Therefore, it has a little green dot there. And we've been able to show comparability in terms of quality, safety, and efficacy. So since we have a lot of scientists in the room, I just want to talk a little bit about the immunogenicity evaluation. Yeah, I've said tech batch 2. It's just a scale at which we were able to lock in the drug substance process at 1 liter. So this is the final batch size that we will use for our drug substance. We did the study in bulb C mice, and we did intramuscular vaccination at day 0 and day 21, also taking bloods before and after, and the experiment ran for up to 42 days, and then animals were euthanized and specific tissues harvested, which included spleens. We measured antibody binding activity, neutralization, and also looked at cytokine responses. And these are the number of animals and doses that were tested. So here we compared Afrivac 2121 with the Pfizer vaccine. We are hoping to do a publication around this. I'm not showing you specific results, but I can give you a summary. And we were able to show that we had dose-dependent binding and neutralizing antibodies. We were able to show that we had similar antibody titers between Afrivac 2121 and Pfizer, and that even at the low dose of Afrivac 2121, that we had slightly higher binding and neutralization antibodies. The CD4 and CD8 cytokine profiles were very similar between our vaccine and Pfizer and Moderna. And we also saw that the Th1 cytokine response was dominant and Th2 minimal. This is also similar to the Pfizer and Moderna profile. And with the cytokine responses, we also saw that those were dose dependent. And this is important because it's very important to do your dose curves. We saw that your dose response curves and your antibody levels would be different to when you look at your cytokine levels. So it's really important that these dose response curves are done. In terms of the toxicology testing, so we were able to show that our vaccine is indeed safe. We had no mortalities, clinical observations were all good, normal body weight and food consumption, slightly elevated body temperatures, but those returned to normal after vaccination, and that Afrivac 2121 did not cause any adverse events. We've recently published the results from the hamster challenge study that we have done. This was in collaboration with the University of Marseille. And so I encourage you to go and have a look at this paper published in Vaccines recently. So just very quickly to show you, we did this in the golden Syrian hamster model, where we compared Afrivac 2121 with the Moderna vaccine. So you can see the dosing strategy over here. In the first part of the experiment, we looked at the immunogenicity, so looking at anti-SARS-CoV-2 IgG. You can see that there's no significant difference between the two vaccines. We also looked at antibody neutralization. So this speaks towards the functional antibodies that are being produced. And then moving on to the protective efficacy studies. So that's the challenge part, following the immunogenicity, again, checking the IgG levels. We then also monitored the viral load in the nasal passages, in the lungs, and looked at weight change over time. And again, no significant differences between the vaccines. So just to summarize that, we saw a good safety profile and comparable in the hamsters, comparable anti-spike IgG levels, and in terms of protective efficacy, protection against weight loss, comparable suppression of nasal viral replication, and also in the lungs, reduced cytokine profile post-challenge, and comparable protection against lung tissue destruction. So, so this basically concluded our preclinical package and validation of the mRNA platform, which gives us greater surety as we move forward with the other disease targets. And so just moving track a little bit, safe to say that, you can develop vaccines, you can build manufacturing capacity, but that alone does not give us sustainable solutions. So what happens between pandemics? So we can develop vaccines, we can design them, we can do all the preclinical and clinical testing, we can take it through regulatory authorization. And then we need to focus on deploying vaccines. So it's not vaccines that save lives, but vaccinations that save lives. And so distribution administration is equally important. But outside of the pandemic, our manufacturing facilities cannot stand idle. So we have to continue to enhance technology. We have to develop products for both regional and local use. And we must also have the regional preparedness. And so this is the beauty of the network that is being established, that in the pandemics that come up in future, that we can collectively respond. And so we did this exercise together with the WHO and the MPP, where we looked at what are the specific regional health needs and what do our manufacturing partners want to be making? And we looked at where are there commonalities across the partner network. These are the ones that you can see here. So you can see that there are some that are common to all, but there are some that are also uncommon. Within Afrigen, we are focused on what we call the big five, not lions and leopards and so forth, but the big five in Afrigen, which is HIV, TB, RSV, Rift Valley fever, and gonorrhea. And these programs are up and running. Some of them have attracted larger grant funding, including by CEPI, but there are others that have only just had seed funding, and we need a considerable cash injection to take that development forward. In terms of the actual mRNA technology innovation strategy, we've partnered with very many collaborators in these specific areas, including manufacturing technology, which looks at continuous manufacturing technologies, such as the system that is made by Quantum called the Intensify. Some of you will be familiar with that. We're focusing on formulation research, so other ionizable lipids, formulations that are thermostable, formulations that can be used in alternative delivery methods, such as microarray patches. We're looking at improving the RNA itself to give us greater durability, to give us larger amounts of RNA, and as I showed you previously, the additional disease targets. So strategic collaborations are really key for building sustainable mRNA platform. And then closing thoughts. So I know that I've gone pretty fast and I've had quite a lot to say. So I'll leave you with these thoughts. We've seen the value of mRNA technology During the pandemic, it has had positive public health benefits and prevention of severe disease and death. But something that is so key is that we need to have global access to these products. And so therefore we have to we've got to keep the momentum, we've got to keep the political will. It has to be a world priority. mRNA technology presents lower barriers to entry, an opportunity for low middle income countries, which we are trying to capitalize now, that we can become self-sufficient and that we can move away from only full finish of drug product, but actually moving towards making drug substance and drug product on the continent. I think it's really important to say that we see that there is a space and value for every other vaccine technology platform. So that includes, vector-based technologies, subunit vaccine technologies, and so forth. They all have a space to contribute. We've seen quite a change from manufacturing facilities that are just built on single products, moving towards multi-product platforms and multi-product facilities. In Afrigen, we've been able to demonstrate that we have a mRNA technology platform, that it can be manufactured in a GMP environment, and it has been validated through preclinical models, and now we are transferring it to low middle income countries. But manufacturing alone is not sufficient for sustainability, and so we have to develop know-how. We have to develop capabilities and capacities for vaccines, and this includes from concept, design, testing, optimization, manufacturing, and clinical development and registration so that we can ensure sustainable access. And lastly, genuine partnerships and mutual support is key through R&D and manufacturing ecosystems so that we can be successful. And so that brings me nicely to this last page, which is really to acknowledge all the partnerships, all the contributions. It has been really a global village in this project. It also has included, some key equipment and technology suppliers who have walked the journey with us. And I also want to acknowledge a dedicated, competent team in Afrigen, and all our collaborators who have worked shoulder to shoulder with us, doing the actual science, doing the R&D, developing the manufacturing processes. We are truly grateful. I will end there. It's on the hour to say thank you very much for your attention, and I'm very happy to take your questions.
So thank you so much, Caryn. That was a very interesting talk. And I think you've covered so many of the elements that would have been questions, but I'll open the floor to anyone who has a question. You can either raise your hand or you can post on the chat, and then I'll help with reading them out just to coordinate for you, Caryn. So open to questions. Please raise your hand or post on the chat.
So we see a hand. Carlos, I see your question in the chat, but please go ahead.
Let me just allow you to speak.
I have actually. You can unmute and Carlos.
Go ahead, Carlos.
Yes, here I am, I think.
Yes, we can hear you.
Perfect. So thank you, Caryn, for such an insightful presentation. It was incredible to see all the work done by Afrigen and collaborators. So very impressive work. I just wanted to highlight one point that you addressed regarding the challenges in the design phase, and you mentioned particularly that for the, let's say, the quality by design phase is still a challenge for the platform for Afrigen. So I was wondering if you can dive in a little bit more on these challenges and how this will be tackled to ensure that the future mRNA products can be, more rapidly available in the development phase. Just to understand a little bit more on these challenges and the quality by design.
Right.
Thank you.
Sure. Thanks for the question. It's an excellent topic to actually discuss. So when Afrigen started the development work, we used COVID-19, as I said. So one of the things that we did was we looked at what is the information in the public domain, and we looked at, what have others like Moderna and Pfizer have done. Okay, so it's a little bit atypical because what you really need to do is to start, as I said, with your TPP, sorry, and then do your risk assessment and go to your critical quality attributes from there. So a lot of the stuff that we have done in a sense for COVID-19 retrospectively. And as we move forward now with the new disease targets, we've learned from that process on how it should be done and how you're going to do it moving forward. So we can now move away from it retrospectively and to be more proactive in the space. I think we're not so the quality by design approach is not so much the challenge that I was referring to. It's actually in the as we move forward and as we understand the mRNA technology with more data that's coming about, how are we going to effectively take that into the space and make sure that as we go in the design space that we are evaluating things in greater depth, making sure that we do the risk assessments for all the different things. I think that a lot was learned from COVID-19, but now as we move into the different disease targets, that we'll be able to tighten that. And another important aspect of it is that what I want to call the sort of phase appropriate approach, right? So a very important aspect that the regulator wants to see is that you have sufficient control. And the demonstration of that sufficient control changes to become more stringent as you go from we're still in early R&D to now we're in phase 1, 2 clinical trials. Now we're going to go to phase 3, because only once you get to phase 3 clinical trial data are you actually able to have where you move away from draft acceptance criteria, but you actually go to specifications. And like a question may be, I'll use an example of like the stability of your vaccine. You may have done indicative stability at the more R&D level, but you actually are able to validate that only in a clinical trial at a later stage. So it's quite a lot of things that we are still learning. We've been gaining experience along the way. I know I've given you a very roundabout answer, but I hope that it makes some sense.
Carlos, are you still there?
Yes, thank you for the answer. And yes, indeed, I guess like the product development advances and the regulators requirements are much stringent. But coming back to just if I may I continue with a follow-up question, as you mentioned the regulation part, how is envisioned? I mean, these modern products, have these all these stringent requirements, but in the mRNA Hub program, there are different countries participating, and that means also different regulator agencies. So how is envisioning, or at least in South Africa or at the African level, this regulatory pathway, because I guess some of the regulators may not be prepared at the technical level that these frontier products need. So how is the the habits tackling that?
Thank you. That's a very good question. So there are a few things that need to be raised with this. So one of the things is that along with the technology development program for mRNA, that there's also been a regulatory strengthening stream, and the WHO has been working with the different regulatory agencies on the continent to upskill them on mRNA technologies. Afrigen's specific experience has been that that while we need to keep an arm's length from SAHPRA, our regulator, that it is also in a way something that we are journeying together with them, because our regulatory agencies have not had opportunities to certify facilities where APIs are being made, number one. mRNA is a new technology, so that's the second thing. So Afrigen has, really been sort of pioneering the space. We've worked together with SAHPRA. One of the things will be that the way that SAHPRA inspects us via GMP will also be an example to the other regulators on the African continent. So it will become a training opportunity for them. An important part of the regulatory system is that in order for the regulatory authority to be able to certify facilities and or vaccines, they have to reach a certain maturity level. So that is maturity level 3. At least, and in South Africa, we've just recently reached ML3 status. We have ML4 for other aspects, but there aren't, we need to increase the level of maturity for our regulatory authorities. And as there is a greater need and a greater drive, and we start to really build the manufacturing ecosystem, it's important that our regulatory agencies are being sufficiently capacitated as well. So I think that, we need to get, some more technical people into that space as well. So there's a lot of activities that are happening in terms of capacitating the regulatory environment. And I'll say just one last thing on this. There's also I think it's the AMA, which is the African Medical Authority, which is going to help us to have reliance across the different agencies on the African continent.
Thank you for that, Caryn. Any other questions from the floor? I can see one hand up. Jeff, let me just scroll to you and unmute your mic. Please go ahead, Jeff.
Awesome, thanks. Caryn, great presentation. I noticed you working quite a lot with Marseille as well as Pennsylvania. Do you anticipate that we're going to have to see more animal model work happening domestically as well as new products being driven through? I mean, there is also that reliance on collaboration for sure, but it'd be great to see kind of more of those capabilities for animal model work to be done locally as well. Have you seen those partnerships developing as a result, or you think they're still to come?
Hi, Jeff. Thanks for the question. I wanted to speak a little bit about that, but I was running out of time. So we are talking about closing the gaps in the value chain and capacitation. So we worked with Marseille, and the reason for doing that is because they had at the time evaluated all the other COVID vaccines. So they'd established the model, they've evaluated, the other commercial vaccines. And so for Afrigen to have them do it also gives everybody a sense of confidence. But with that, we also were part of connecting Marseille with the University of Cape Town, who have now, through the collaboration with Marseille, established the hamster model here. and there will be some challenge studies that will happen following that. So that's one example of it. For the GLP toxicology study, we worked very closely with Northwest University's preclinical development platform to evaluate what we can do and where the gaps are. For Afrigen study, we used an Indian company called Agile Life Sciences, and through that process, Afrigen is also able to work with Northwest University to further capacitate their sort of GLP toxicology hub, which is being established. So that will enable us to be, to run the full GLP tox here, not only housing the animals, which we can do very well, but also including histopathology, hematology, and so forth. So essentially a one-stop shop. In terms of the work that we've done with the University of Pennsylvania, we have very close collaboration with them. And the great thing about it is that there's such a willingness to be able to transfer the tech back here. So even where I spoke about the dendritic cells and looking at reactogenicity, we were able to then establish that in-house here. So there's a lot of that work going on. And as we move into the different disease targets, certain diseases have a higher level of biosafety, so you need BSL-3s and so forth. And yeah, so on a case-by-case basis, but trying to bring everything on the continent into South Africa and to partner with our the different partners in Africa. So there's a lot of capacitation work that's going on. But yeah, I wish that we had more days, more hours in the day, and that we had, a much bigger budget to work with.
Oh, awesome to see. Glad to see that the tech transfer was coming back. Cool.
Thank you.
Thanks for that. And I have a question from Nicole Johannes. She says, thank you for the presentation. What do you think the timeline will be when we would be able to see complete manufacturing on the continent, not just fill and finish?
Wow, well, that is the big question that everybody wants to know. So there are very many initiatives that are happening on the continent at the moment. So as I was saying, as part of our tech transfer program, Afrigen has transferred to Biovac. So they are the South African commercial manufacturer. So many of the manufacturers that we are tech transferring to have used COVID-19 as a platform, but they will not take a COVID-19 product to the market. So that means that the next disease targets will need to be the ones that we take forward. I can only really speak to the ones that we are currently working on. So some of them will take a longer time. So let's talk about TB, for instance. Like you need to get into arms very quickly, and you have to have a different strategy because we don't actually know the correlates of protection and so forth. So in order for there to be manufacturing and commercial manufacturing, we need to have develop the technology, and it has to go through all its regulatory processes, but you also have to have the demand. So it's not a straightforward question. You have to have the demand. Your local governments have to say, yes, we are prepared to purchase those vaccines. So it's a lot of market shaping that has to happen. Very many things that have to come into play. I think that in South Africa, we're on a good trajectory to be able to be doing complete manufacturing within the next, I'd say 3 years, 2 or 3 years. There are other initiatives that I can't speak to in great detail, but there's BioNTech initiative in Rwanda that's come online. There were some mutterings about the Moderna setting up in Kenya, but that has been halted. So unfortunately, it's very difficult for me to say the exact timing, but I think within the next 2 to 3 5 years, we should see that we have complete manufacturing from drug substance to full product in a few of the manufacturers that we're working with.
Thank you. And I think Nicole is happy. So any other questions from the floor? I'm trying to see. I can't see any hands up, and I can't see anything on Q&A. Let me just quickly check the chat, and there's nothing on there either.
There's
So
on Q&A, you could check one more question there from Leiva Carlos.
I think Carlos, it's the same question he had asked
Okay. Makes
verbally.
sense. All right.
Yeah. So I can ask a question if you don't
Please.
mind.
Sure.
So you presented that you were able to establish quite quickly, and that you had personnel readily available. Would you say that this was unique to South Africa, or this is something that we would expect would be mirrored within the continent, or would there be efforts to try and help strengthen capacity in the different science fields and levels of learning that would be required to support localizing manufacture in other countries?
Yeah, so that's a good question. So I can say I think that what we've experienced here in South Africa would be quite similar to some of the other countries in Africa. We had a rich pool of people, particularly in the research and academic space. But we struggled with finding people that actually have the GMP know-how. And that is a symptom of us not actually having a biomanufacturing ecosystem, or actually it's, biomanufacturing is very small, particularly in South Africa. So even of the people who we could have access to working in pharma, is very small. People rotate to the very different, to the different companies like Aspen and Biovac and Adcock. And so, very small pool to work with. We did not see a problem with a technical and scientific background, but the challenges around understanding quality systems working in a GMP environment, that's where it's been quite difficult. I think there's considerable training efforts at the moment, which include the WHO program, but many other programs that are around training to get, the quality side of things installed, and capacitation of that. And I think that there's very much to be said about actual hands-on training, and part of the technology transfer process is, also about transferring the tech, but also hands-on training. So for us, I think the two key things, Marianne, was understanding and knowing quality systems and how to work in a GMP environment and in a quality controlled environment, which includes a lot of controls and documentation. And then the second thing for us where we really struggled was on the formulation expertise side. So there's a lot of formulation capabilities in pharma, which is around solid drug dosages and so forth and so forth. But when it came to sort of encapsulation technologies and more liquid technologies, that has been lacking on the continent. And so we actually had to develop that capacity in-house, and work with collaborators and train ourselves to get up to speed on that. So I think that there's been an estimation or some exercises in terms of how many more people we need to capacitate biomanufacturing on the continent. And we are quite far behind on that. So there needs to be a considerable increase in training and capacitation in the biomanufacturing space.
Okay, thank you. That's good to hear that at least you were involved in the training, and that's something you're working on. And I think, as you said, that's something that would be mirrored across all, and is more of a feature of lack of active manufacturing in human vaccines within the continent. Right. So I'll move on to Ignacio. I can see you've posted a question. Would you like to speak to it, or are you okay for me to just read through it? Okay, I can't see a hand. Oh, there you are. Let me just unmute your mic. Please go ahead.
Thank you so much. Hi, Caryn. Thank you so much for the presentation. I apologize because my question is related to Nicole's question, but maybe just to give the chance, is it great, the explanation of the timelines for developing a vaccine, but what do you manage, for example, in a pandemic outbreak? Maybe for that you need to respond very fast. So it could be nice to see how do you manage these situations? Thank you.
Thanks. That's a great question, and it's a question that we've been discussing in Afrigen, like how quickly will it take us to convert the existing activities in Afrigen into pandemic response mode. I don't want to throw out a number yet because that may it may cause a lot more other questions. But I think that within I think it's around the 20 to 30 day in terms of converting the facility into pandemic response mode. That is what we are looking at in Afrigen. Obviously, not just converting the facility is important, but also, well, how quickly can you then go to take that particular disease target and put it into the mRNA platform? And that is something that we're working quite hard on. I think that CEPI have and a lot of others have the 100-day approach. The difficulty with the 100-day is that, we can possibly go to getting first vaccine candidate made, but that excludes the analytics part of it. So I've thrown out some numbers now, 20 to 30 days to put the facility into pandemic response mode, 100 days to get, the first vaccine made, but then we have to do testing. So I can't give you a precise date at this stage, but those are the numbers that we're looking at the moment. And we are looking at reducing those numbers, like I spoke about, using synthetic pDNA versus conventional plasmid DNA, and there we see that you can reduce your manufacturing process by I think it was from 6 to 8 weeks in the case of pDNA to 2 weeks for synthetic DNA, and these are things that are all currently being tested and validated with our partners currently. So I know I want to tell you that we can produce a TB vaccine tomorrow, but it's still going to take us some time. And in the case of the next pandemic, we're hoping that, we're shooting for the 100-day mark, but we still have to take into account analytics. And one of the key things is, can we do the analytics faster? So we welcome any solutions and collaborations in that space.
Thank you, Caryn, for the explanation. And congrats again for the work you are doing at Afrigen.
Thank you.
Okay, so I can't see any more hands raised. Oh, buzzer beater, Terence. Let me just unmute you. There we go. Sorry, Terence. Are you able to speak? You can unmute yourself. Yeah, there we go.
Okay, thank you. Hi, Caryn. Thank you for a great presentation there. It was lovely, and just to see how much you've done, the kind of work that you've gone through to get to this level. That's really, really, really great. Then my question, I just want to delve into, because you are building a capacity for mRNA development in terms of the collaboration with the institutions which are working with zoonotic pathogens. How strong is the collaboration so that you can have a good surveillance on what is happening on the ground? Because a lot of researchers, they are working in that space, but if ever there's no connection there, you might be missing a lot of things. Then I just want to know how strong is the collaboration, and if not, then are there any plans to enhance them or to develop them so that you can have a good surveillance on what is happening on the ground? Thank you.
Right. Thank you, Terence. That's a great question. I think that those of you who are working in a surveillance space will know that during the pandemic, South Africa was actually really instrumental in surveillance. We had established quite an extensive genomics surveillance platform in South Africa under Professor Tulio D'Oliveira. So what Afrigen is trying to do is not to reinvent the wheel, or also be working on that, but to really have effective collaboration, which is, I think, what you're asking. So just to say that that Tulio through his program has, I think, helped to establish quite an excellent genomic surveillance network on the African continent. So we are trying to plug into that. I can give you 2 examples of this. So on the sort of zoonotic side, so our Rift Valley Fever project, which I know Kenya is very active in the Rift Valley Fever space. But so we were looking at incorporating the Rift Valley Fever antigens into mRNA. And what we did from that is actually to look at consensus sequencing, but also from the consensus sequences to also look at how they compare with specific outbreaks in Rift. And we did that in collaboration with Tulio and his team. We'll do this in the case of monkeypox. So now we know like what circulating strains are happening. And if we're successful in the grant application that we've done for mpox, that one of the key things will be to look at the data that we've got from the genomic surveillance and make sure that we incorporate that into our design for our mRNA constructs. So yeah, there's definitely a concerted effort to make sure that particularly in the zoonotic space, that we're working very closely with the networks in genomic surveillance. And I think that we can see a bigger increase in that happening with those that are developing other vaccines and using other platforms as well. So I hope that answers your question.
Are
Yes.
you good, Terence? Okay,
Oh,
great.
yes, I'm good. Thank you, Caryn, for that.
Yeah, so I think we're at 2 minutes to time now. So if there's no urgent burning question, we'll thank Caryn for your time and for the wonderful talk. And it's been very interesting, and hope to engage with you further with our own initiatives, possibly with our upcoming vaccinology course, but we'll be in touch. So thank you everyone as well for your time and attention. And I will end the webinar now.
Thank you very much, everybody. Thanks for listening.